Hello, good morning. My name is Sue Charman-Anderson. I'm the founder of Ada Lovelace Day, and I'd like to welcome you to the second Ada Lovelace Day 2020 webinar, in which we will be asking what are medicine's biggest questions. With me today to discuss this are Dr. Freya Harrison, Dr. Mariam Jamal-Hanjani, Professor Ijeoma Uchegbu, and Dr. Jen Edwards. We will have time for questions later, so if a question occurs to you as our panelists are talking, please do leave a comment. Facebook or YouTube, doesn't matter, and we will have a Q&A at the end. So thank you very much to our panelists. Thank you very much to you for being here, and I will let the panelists get on with it. Our moderator for today is Ijeoma Uchegbu. So thank you very much, Ijeoma. Take it away.
Thank you so much, Sue, and welcome to all of you that are watching this webinar on medicine's biggest questions. And we're going to start off with introductions of the panel. And my name is Professor Ijeoma Uchegbu. I am a professor of pharmaceutical nanoscience, and that means I'm interested in very small things. And I work at University College London. I'm also the Pro-Vice Provost for Africa and the Middle East at University College London. And I'll go over to you, Dr. Freya Harrison, to introduce yourself.
Hi, thanks, Ijeoma. I'm an associate professor in microbiology at the University of Warwick, and I work on bacteria.
Brilliant. And over to you, Dr. Mariam Jamal-Hanjani.
Thank you. I am a cancer researcher at the UCL Cancer Institute, and I am also a lung cancer oncologist at UCL Hospital.
And then over to you, Dr. Jen Edwards.
Good morning, everyone. I'm Jen. I'm a University Academic Fellow in Musculoskeletal Medical Technologies at the University of Leeds. And it's basically, I'm a lecturer, but I have a bit more time at the start to dedicate to my research.
That's absolutely fantastic. And, you know, I guess our viewers would like to know, for example, what's been our journey into science? And so I'll start off with mine. I mean, I sort of stumbled into pharmacy. That was the only course that looked vaguely interesting when I was going to university. So I ticked that, not knowing at all what it would entail. And then eventually I did a PhD in pharmaceutical sciences simply because I didn't want to have a patient-facing role. And then since then, I've absolutely loved it. I've got a research group, and my journey has not been straightforward because when I was doing my PhD, I was a single parent actually living in a bed and breakfast for the homeless. But you know what? If I can do it, any of you thinking of being a scientist, you can do it too. And now I have a great research group and we're doing interesting research. Mariam, tell us about your journey into science.
As a child, I was always fascinated by the natural world, and I really liked the way that we could use concepts in physics and mathematical language to describe and explain how the world worked. And I don't, probably not like almost every child, but I think many children want to be an astronaut. So did I. And in fact, I studied physics as my first degree and really enjoyed it. But in my early 20s, in search of a vocation that perhaps I felt was more fulfilling for me personally, and one that met my interest in human physiology, I studied medicine. And in medicine, after getting my medical degree and training, I always wanted to go back to science. I come from a family of mathematicians, and I always wanted to go back to science. So oncology allowed that. And in oncology, it's expected that you do a PhD and you do research. And the wonderful thing is you can do basic science research or clinical translational research. So that's how I am where I am today.
Fabulous, Mariam. I love that. A family of mathematicians is brilliant. Freya, tell us about your journey into science.
So I actually chose my A-levels not thinking about becoming a scientist at all. I wanted to work in the music industry, but I'd been brought up by parents who were very keen on conservation and botany in the natural world. And so out of general interest, I decided to also do an A-level in environmental science, and I loved it. And I knew within the first half term that this was what I actually wanted to do. I wanted to do something that involved problem solving, that involved answering questions about the natural world. So I did a 180. I picked up an extra A-level in biology. I went to university, and again, I knew that was exactly where I wanted to be straight away. And I decided fairly early on that I wanted to do a PhD, probably inspired by my third-year research project at university of finding out what it was like being part of a lab team.
Oh, wonderful. That sounds really good. So you start off almost thinking about botany and then decide, well, actually, I'm going to go the whole hog with biology, and yeah, then you end up where you are. I think that's brilliant. Jen, tell us about your journey into science.
So I guess if we're going back that far, I grew up on Tomorrow's World, Star Trek, and David Attenborough. So it probably explains a lot. But I knew I enjoyed science, so I did all the sciences and maths at A-level. I decided I wanted to go to university and do physics because it was cool and nobody was doing physics. And then I failed catastrophically in year 2. And I decided that that was a combination of my personal circumstances and physics just not at degree level not being for me. So I ended up doing artificial intelligence and computer science, and whether my cat is in the background. And my third year project made me realise, actually, I missed biology. So I applied to do a PhD where they wanted physical scientists to work on life science projects. Then did a degree in cell biology and mechanical forces. And I ended up working as my first postdoc in Leeds, looking at replacements for natural tissues, and that's where I've been ever since. So it did definitely not go to plan in the first place. I didn't really have a plan for it to go to anyway, but here I am, and I seem to be doing all right.
Oh, it's incredible how it's so varied. We've got people that sort of were quite keen that they wanted to be scientists when they were quite young, and then we've got your story, Jen, where you started and it was sort of, you had to stop and almost sort of recalibrate, and also Freya, your story about, you know, you botany and then moving into biology, and Mariam, the fact that you've got all these brilliant people, role models in your family, that is really, really cool. I mean, it's so varied, and you know, my journey, you know, looking after young children doing PhD, it just shows you that the route to being a scientist, there is no traditional route, actually, definitely not for us women. I mean, would you agree, Mariam? There is no traditional route.
Absolutely. And I think that your family background, your personal circumstance, everything molds the decisions that you make in life. I think as long as you're driven and you're passionate about what you want to do, hopefully you end up in the right place.
Brilliant. And so now we're going to sort of talk about the research we're all doing so the audience gets a better flavour of the kind of things that we're interested in. And so I'll start off, and I'm really interested in developing new medicines. And what we have done is that we've taken a hormone that's in the body. I mean, you could call it a hormone, and we've made this into a medicine. We've made it into a pain medicine. So we've taken enkephalin, and that's something that is secreted in our brains when we feel pain. And we've used that to make a pain medicine. And we've out-licensed it actually to a company in the United States. And they've managed to get the NIH to take on, the NIH is the funding body for medical research in the US. They've managed to get the NIH to take on completely the development of this medicine. And it's happening at the moment. And so it's really, really cool that next year we could actually be going into humans with our particles containing enkephalin. And the reason we did that is we know that a lot of people who have severe pain, they take drugs like morphine and fentanyl. But these drugs are actually quite dangerous, because if you go over a certain level, you do then end up with a difficulty in breathing. And so what we find is that in the US, 15,000 people on prescription opioids, that's morphine and fentanyl, actually die because either overdoses or just taking the drugs and ending up, you know, taking too much, because these drugs are also habit forming. So looking at that, we saw a real problem, identified what we felt was a solution, and then made this new medicine, which is now being developed. So it's quite exciting. I hope it goes all the way, you know, because as well as being exciting, there's going to be a little bit of this as well. So, you know, it really is something, you know, we've worked all our lives doing this and spinning out a company to get it done. And that's been quite a difficult journey. So Freya, tell us a little bit about what you're working on at the moment.
So my team is particularly interested in bacterial infections, and especially the really long-lived chronic bacterial infections that tend to affect people whose normal defences are compromised in some way. So people who have the genetic condition cystic fibrosis get these basically untreatable infections in their lungs. Their lungs fill with sticky mucus as a result of the genetic mutation. It's a brilliant environment for bacteria. Once they get in, they can't get out. Also, people who have diabetes get infected ulcers on their legs and feet. They don't respond to antibiotics, and very sadly, people end up having to have their leg or their foot amputated, otherwise there's a risk of the bacteria triggering sepsis. And what we want to do is to really understand what bacteria are doing inside these infections that makes them impervious to treatment. They behave really differently than they do in a test tube or on a Petri dish. And we normally study bacteria in test tubes and on Petri dishes. And we normally try and develop antibiotics by testing them against bacteria in test tubes and on Petri dishes. And so what we're trying to do is to build and use much better models of what bacteria find inside a human body in these infections. So one thing we're doing is that we go to our local butcher and we get pig's lungs that are waste from the meat industry. And we and our collaborators have a way of cutting out small sections of pig's lung, growing them in laboratory medium that really mimics that sticky cystic fibrosis mucus, then infecting them, seeing what the bacteria do. And that helps us to do some fundamental biology work. I'm hoping it'll help people replace live animals as well. But we've actually just also worked with a company to have that accredited as a testing method for pharmaceutical companies to use against bacteria. And we're also trying to develop new ways of treating these bacteria in these lung infections and wound infections by looking back at historical and traditional medicine, looking at the plants and materials people used in the medieval period, and using that as a database for natural compound discovery. And we're hoping to go into initial human safety trials next year with one of the preparations that we've found.
Oh, that's wonderful. And how closely does pig's mucus mimic the infectious mucus that cystic fibrosis patients have? Because that tends to be stickier and more viscous. So how closely is healthy pig mucus resembling that?
Well, interestingly, the pig lung secretions are more similar to human lung secretions than, say, mice, which would be a standard lab animal, are. People have done sort of NMR and mass spec on the small molecules in the lungs, and they're more similar. But we actually make a synthetic version of cystic fibrosis mucus. So my collaborator Marvin Whiteley in the States actually developed this by looking at the chemistry of CF mucus, so we can make it in the lab.
Oh, that's fabulous, because that is a big problem when it comes to gene therapy and cystic fibrosis, the mucus. So yeah, that's brilliant. Thanks, Freya. Mariam, tell us a little bit about the research you're doing at the moment.
My PhD was in cancer genetics, and I treat patients with lung cancer. So it's quite natural for me to want to try and understand lung cancer as a disease. The way we historically have studied cancer has relied on cell lines, animal models, but really to the wealth of information you get from tissue collected from patients or blood samples is very different, and gives you a real insight into the disease process. So quite a few years ago, with then my supervisor, Charlie Swanton, we set up a study called PEACE, which is a national research autopsy study. And in the beginning, we thought that this was somewhat controversial, and there is a real stigma around discussing death amongst my peers, so fellow medical professionals. But actually, patients have been our biggest advocates, and this study has been recruiting UK-wide. And the real aim of the study is to understand why and how cancers evolve. So a tumour evolves as a result of, say, the treatments that we give our patients and other things that they may be exposed to, what we call selection pressures. So using research autopsies, patients donate tissue and blood after they die. And we are able to collect fresh tissue samples from where the cancer has spread to different parts of the body. We're trying to understand the metastatic process of disease dissemination. We're trying to understand what the mechanisms are. Is it all about cancer evolution? Is it the cancer genome? Is it the genetic landscape? Is it the environment in which a tumor exists, the so-called tumor microenvironment? Is it the patient or the host immune system, a failure for the immune system to recognize those cancer cells as foreign entities? Is it something about the rewiring of the cancer cell metabolism? So we're trying to understand the metastatic process and ultimately why patients die of their disease, given that there is a real unmet need. The main cause of mortality in cancer patients is metastatic disease. If we can try and understand those processes in end-stage disease, we might be able to slow it down, possibly prevent it. So there's a real unmet need to try and understand why our patients develop metastatic disease and die. And research autopsies have the potential to transform this area by virtue of being able to access tissue.
That's brilliant. And I wonder, Mariam, when you're looking at cancer patients at autopsy, but I've heard that even so-called patients that have died from other causes, when they're actually examined, they're found to have multiple tumors as well, but which haven't caused them any difficulty. And what's the new news on those kind of patients?
So that's really fascinating. In particular, for example, if you look at normal lung, I say normal, so lungs that on a scan looked as if there was no disease there, you can find what we call pre-invasive lesions, or lesions that could have turned into tumor but somehow have not, and like you say, have not caused the patient problems. Or you may find other synchronous, simultaneous tumors at autopsy. And we don't know if it's perhaps the immune system that's controlled that disease and not allowed it to disseminate. We don't know if there's something about the microenvironment, whether it's the vasculature, whether it's the stromal cells that are surrounding the cancer cells that prevent it from progressing and disseminating. So there's a lot we still don't understand about that. But absolutely, there are incidental findings at autopsy that would not have caused the patient any problems when they were alive. So there's a lot more we can learn beyond the disease that we were aware of before the patient died.
Fascinating, absolutely fascinating. Thanks, Mariam. Jen, tell us about what you're working on at the moment.
Sure. So my research is around regenerative therapies in the foot and ankle. So a lot of the work that I do relies upon the idea that if we've got a damaged tissue, often the best thing to replace it with would be more of the same, but we don't have spare parts. And you get issues with donor tissues in that the cells from a donor would be recognized as non-self, as foreign, and your immune system would attack them. But the idea is that actually for a lot of your tissues, particularly things like ligaments, which have a biomechanical function, it's not the cells that are the important part. It's the matrix of collagens and different proteins that they build that's the important part. So we've got a process that essentially washes out the cells, leaves the matrix behind, and leaves it mechanically functional so we can use it to replace damaged tissues. So I'm really focused on replacing ligaments or tendons to look at treatments for chronic ankle instability. But actually, interestingly, Freya said before she's working on diabetic foot ulcers because they're problematic. And I'd like to help prevent them getting to the point that the ulcer forms. So one of the problems in the foot is that the fat pads on the heel and the ball of the foot can degenerate over time. And especially in a lot of diabetic patients, they become very thin and sort of flattened. And because diabetics also often have ischemia, so they can't feel when they've got, say, something in their shoe, they'll get a pressure point. They've got abnormal load biomechanics because that fat pad's gone, and they get a point of high pressure, which is where the diabetic foot ulcer forms. They might not notice because they can't feel any pain in their foot. So what we're trying to do is develop a solution that we could use to inject into the foot and try and restore that fat pad, not necessarily to prevent diabetic foot ulcers entirely, but just to delay them, because the effects on patient life are so severe, and the 5-year survival rate is terrifying. But you know, it's that thing of, can we repair some of the tissues before we get to these problems that we see, either as a consequence of disease or just as aging. We get old, we damage joints, we damage tissues. And it's about allowing people to be healthy for longer in their lives. Our tagline is 50 active years after 50. You know,
I love that because I'm just shy of 49 myself. But that's brilliant. And when you talk about injecting cells into the fat pads, are you injecting just adipose cells? Are you injecting a particular cocktail?
So it's actually not injecting the cells. It would be injecting adipose matrix. So what we do in a lot of our treatments is assume that if we put the right tissue in there, the body, and we've seen this in some of the animal studies we've done to show that they're safe, is that the patient cells will basically move in and repopulate the tissue. Previous people in my group have had great success in early trials with heart valves. I don't work on those, but the cool thing about heart valves is that you can use a small heart valve for a smaller patient, and instead of them having to have repeated treatments over the years to give them a new heart valve when their heart's grown, their cells move in, that heart valve then grows with them. So it's regenerate the body rather than just repair it, but use the body. All we do is restore the mechanical function and the body will sort itself out.
That is absolutely brilliant. It's just a different way of thinking about transplantation almost. It's a different way, you know, you're seeding the new organ, as it were, the new piece of tissue. That's great. That's cool. So let's move on to the next topic. And we've all talked about our research, and you know, whenever we're talking about our research, the thing about it is that our faces all light up and you can see the pleasure that we get from doing our work. But I know it's not all pleasure. I know that there must be hurdles. I've definitely had hurdles in my work. And one of the biggest hurdles that I actually had wasn't what most people say is, well, getting my first grant was hard, but it wasn't really hard for me. I mean, I remember when I first started out, I wrote 13 grant applications in 1 year, and I got one funded. So I obviously didn't know what I was doing, but I was busy doing it, you know, not knowing what I was doing. But I managed to get one. And to be honest, I didn't know whether I'd get any at all. So when I got one, I was actually quite happy. And the next year I got another one. So that I didn't really find that hard. What I found a huge hurdle to my work was when I wanted to do translation, when I wanted to take my work out of the laboratory and try and see whether it had an impact on people. And so we spun out our company. And when we spun out our company, the biggest hurdle was actually getting that funding to be able to do clinical work. It's so hard. So you've got a great idea, you've got great technology, you have to match that onto societal need. That was hard. Take my technology and make sure that society wants it. Then you have to make sure that society is willing to pay for it, or someone in society is willing to pay for it. Pay enough that people want to do the clinical trials. Then you have to convince the money men to put money in so that you can do the clinical trials. If you get my point, that I found was the biggest hurdle in my work. And I'm going to now ask you about some of those hurdles. We have overcome the hurdle, as I told you, by licensing it out to a US company who then went to their grant awarding body and managed to get, and at the moment it's about $6-7 million is going into just this early stage. And by the time you get into the clinical stage, I mean, we could go up to $50 million to get it into approval minimum. That's just the minimum, you know, because there are certain things that we've already worked out. But normally drug development is $1 billion affair. It's very, very unpredictable and normally unsuccessful. So getting this far, we feel quite a hurrah moment. I'm going to ask you about the kind of hurdles. They may be different from the one I've described. But Jen, tell us about the type of hurdles that you've experienced, even if you haven't solved them. You know, what are some of the biggest ones?
So I think most of the hurdles that I've found actually come down to the biggest one for me, which is my own confidence and my own certainty that I am where I should be and that I know how to do this. And I'm finding it's a very slow process in sort of learning to get past that. So little things like I will suddenly find I'm talking to a colleague and I'm actually telling them, you know, I've got a reasonable answer and I know I've learned something and I can be confident and say that. But it means an impact on things like going to conferences and meeting people who might be collaborators. I find that very hard, which means compared to some of my colleagues, I don't make new connections very easily. So my network for collaborative research is quite small. I'm also very multidisciplinary. So I work almost a 50-50 split between our School of Biomedical Science in the Faculty of Biology and the School of Mechanical Engineering in Engineering and Physical Sciences. And it means that, you know, I'm trying to be very multidisciplinary, and obviously my background with computer science and biology PhD were. But again, it means that you never quite feel comfortable being an expert on anything that you're supposed to be an expert on, which is sort of, I think that, you know, the way I'm coping with it is talking to my colleagues, getting them, I'm not saying getting them to reassure me, but making sure I listen when they say, thank you, that was good, or whatever. Making sure I do the same for them, because one of the things that I've learned actually is this is a hurdle for an awful lot of people, is just that idea that why are people giving me money to do this? Why do I know anything about this? Because we forget that, yeah, we do. We know a lot about this. We've spent a lot of time learning about this. We also all accept that we don't know everything. We wouldn't have jobs if we knew everything, because what we're trying to do is learn. And I think one of the biggest pieces of advice I ever got given from my previous boss was, one of the things that makes us very good scientists is that we question things. We are critical about the literature that we read, the experimental results that we've got, and we do this. The problem comes when we apply that to ourselves. Because when we apply that to ourselves, there's no one there to sort of rein us in. There's no internal filter. You're just very critical about what you're doing. And that's the thing that you have to get over is, yes, you know, think about what you're doing, think about being critical of what you're doing, but also put it down and allow yourself to realize, but I've done this, 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 and this. So yeah, I think my biggest hurdle is me.
I think what you said will resonate with so many people. Yeah. I mean, we have this persona of, you know, you have to develop a persona of confidence and competence, but we're all filled with doubt. And I love the way you've said that, you know, what you then do is when people actually praise you, you actually accept that, and that helps you, you know, go forward. I think that will resonate with a lot of people. And if you wanted to give someone who had a similar feeling about themselves a little bit of advice in a couple of words, what would you say to them?
I would ask them if they would ever say what they're saying to themselves to somebody else, because that is the biggest thing. Is if someone came to you and said, oh, I'm really sorry, I haven't got this done because I've had this happen and this happen and this happen, what would you say to them? Because the answer is probably go and have a cup of tea and sit down and come back tomorrow. You know, you've lost a day, fine. What you're saying to yourself is, oh my God, I have to get on, I have to do this, and I have to be this right this second. And no, so always when you are having those doubts and you are feeling like you're your own worst enemy, ask, if someone came to you with the same problem or the same feeling, what would you say? Honestly, first, what would you say without thinking? And then it is hard to do, but then try and let yourself accept that answer rather than what you're telling yourself.
I think that's brilliant. So what you're actually telling people is that actually most people are not super judgmental and they actually realize that we're human beings. That's fascinating. It's really, really good advice. It's advice I'm going to take next time I feel like
Right, I've done something useful.
Mariam, tell us about the biggest hurdles you've experienced in your professional life.
I think probably the number one is saying no. I'm really bad at saying no. And some of what Jen has said also, I'm incredibly self-critical at a point where sometimes it can be obstructive. But I've got a real way of, in my mind, talking myself around and saying, no, that's not right. You know, you have this ongoing conversation in your head with yourself where you know, or you deep down, you believe that you're capable and you know you're motivated and driven, you've worked hard and you deserve to be where you are. I think what doesn't help, and we might come on to this again, is my advice is always surround yourself by people who are challenging, realistic, but supportive and who know you. So people who are invested in your well-being. It could be that they benefit from it. There's nothing wrong with that if you benefit too. As long as it's a symbiotic relationship, I think that's fine. But I think in not saying no, I take on an awful lot, have done over the years. So I've tried over the years as the research has got heavier, and there's more to do, and there's more to learn. I think I've tried to be better at compartmentalizing. And I think otherwise it's the person you are and your background. So as Jen said, she's split in terms of her role, and I have clinical commitments. And that's a tough thing to juggle sometimes, I find. The demand from, I went into medical school because I know it sounds trite, but I wanted to do good. And I did want to be patient facing, and I enjoy it. And I really have a deep love for medicine and clinical practice and clinical acumen. And I always want to emulate those great clinicians that I met when I was in medical school. But I love research too. And for research, I feel like it's a thing I'm doing for me, but the clinical work is the thing I'm doing for others and what I was made for. So there's always that, I'm torn between those two worlds. And I think you can't master both. I think you just need to be reasonably good at both, cause no harm, and try to find a happy medium where you're fulfilled and you deliver on the research and the clinical work. So I think it's the balance between those two. I suppose it's the balance of workload, which we will all face regardless of the work we do.
Mm. So that's interesting. And I'm wondering, Mariam, do you find it difficult to say no? Because like me, there's a fear of missing out, and you just don't want to miss this next opportunity that's coming your way.
Yes, I think so. I mean, I'd love to become that person who thinks, I don't mind if I say no to this, and it could be the next big thing, but I've already got enough going. I guess we don't feel if we felt we've got enough going, then we wouldn't keep doing the science, I suppose. Yeah. But yes, I think it is. There's a bit of FOMO, as they say.
Oh, that's excellent, because I'm sure a lot of people feel that way, especially when you're starting in your career and you think this is going to be an opportunity I can't afford to say no. It's really good. And Freya, tell us about some of the hurdles you've had to overcome in your research career.
Well, both Jen and Mariam's answers have really resonated with me, actually, and I'm hearing echoes of things that some of my best mentors have told me, when to say no to things, not to try to be perfect at everything. And I think for me, that all comes together in just the cognitive load of the job we do being so huge day to day. Like Jen, my work is very multidisciplinary, so I'm trying to keep up and make sort of contributions to discussions in bacteriology, in natural product chemistry. My work on the natural product antibiotic discovery involves working with people in the humanities and arts, so you can feel very pressured to sort of be knowledgeable and up to date in lots of areas. And if you think about the different facets of the job we do, that can be very demanding. I need to be a creative person in order to be a researcher. I need to be a good manager because I have postdoctoral researchers and PhD students who sort of need me to be their mentor / boss. I need to be a saleswoman to a certain extent, because as you were saying, Ijeoma, you're taking ideas that you want to be translated to industry and trying to get those funded and trying to get those spun out. Because undergraduate teaching and tutoring is part of the job as well. You're also very much pastoral role, and the students can see you in a slightly parental role from time to time, and that can be very demanding. So it's this sort of constant nibbling away of different bits of you and different bits of your strengths all the time. And it can be very, very difficult to sort of sit back and think, actually, I'm doing these things. I am getting everything done. So yeah, that for me has just been the sort of preventing myself getting to the point of mental exhaustion by trying to do all of these things at 110%. And I think something just really fundamental there, as Mariam, I think, said, surrounding yourself with good people. And as Jen said, making the right links with people. Knowing when to ask for help is something I think I found very, very hard in the first few years of my career, and I've got much, much better at. And actually the things that have kept me going are reminding myself that I am actually doing good. I am doing useful things for people, even if they're only small things. So this is why actually I have behind me on the wall underneath Dolly Spartan is an email a member of the public sent to me. So some of my work on the antibiotic discovery got some press coverage a few years ago. And I had this lovely 2-line email from a lady who said, My two daughters both have cystic fibrosis. They culture Pseudomonas and Staphylococcus. I see that your lab works on both. Thank you for your work in this area. And that to me sort of makes it all worthwhile and reminds me that yes, I am doing some good somehow.
I think that's brilliant. And so, you know, Freya, you've actually very neatly described the academic condition. How do you prioritize? How do you determine what's going to take your time in a particular day, you know, when you've got so many competing demands?
A very good question. And if anyone knows the answer to that, I would be very, very happy to find out. I think, you know, my current mentor here at Warwick gave me some fantastic advice. And he said, when you're marking undergraduate work, 70% is a first-class mark. What can you do 70% on and still get a first-class return? And I think that, for me, has been crucial in prioritizing what can be done in a limited time well enough, what actually needs time setting aside to do in great detail. I think that for me personally, is somebody relying on me to do something to enable them to progress? And if that's the case, that's the thing that goes to the top of the list.
Okay, that's very, very nice. I mean, a lot of people only think about themselves, to be honest, because if you don't think about yourself, who will? But it's nice to hear from someone who's also thinking about mentoring and students and people that need references and all sorts of things. I'm going to now talk about funding, and that the funding, of course, is very important. If we don't have money to do research, we can't do the research. So we're all talking about our research, which means we've all been able to secure funding. And I think, you know, thinking about how we fund our research, I've had unbroken funding from the EPSRC since I got my first grant in 1999. I think actually got it in 1998. And since then, I've had unbroken funding. But I've found it harder and harder and harder to secure funding. It could be me. I could have hit peak ideas and now I'm sort of moving out to pasture, or it's just become more competitive for everyone. But it's really, really difficult to get funding these days. I mean, I chaired an EPSRC panel earlier in the year, and I think we kind of asked them, how many people will be funded? And, you know, they never want to give you an answer. But, you know, doing the maths, it looked like only 10% of people that got to the panel, 10 to 20% would be funded. So 80% of people who got to the panel and maybe 90% of people who applied, I'm just thinking of these numbers, probably they don't get funded. In the early days, the European Research Council said when they first started these ERC grants, these are grants that are awarded, the EU used to award grants to collaborative groups. Then they started awarding grants to individuals, and those were these ERC grants. The first time they came out, which was about 12 years ago, the success rate was 3%. So 97% of the proposals which you work on, most people work on their proposals for 6 months at least. 97% of those don't get funded. Sometimes you wonder, are we spending our time wisely here? But I'm going to ask you about funding. And I'm going to start with you, Freya. Tell us about how you fund your work. Has it been easy? I know the answer is no. How difficult has it been?
So there's
funding.
There's definitely that lag phase as you begin your career when no one will fund you because you haven't previously been funded. They want you to have proven yourself before they give you the cash to prove yourself. And sort of once you get over that hump, it suddenly gets a lot easier. I guess I'm in quite a lucky position in one respect, in that the work I do is not expensive. We don't require extensive facilities, beamline time, etc., etc. But where things have been very difficult, and I think Jen might agree here, is when you're trying to get interdisciplinary work funded. So, and this is a big buzzword, all of the funders say, we want interdisciplinary work. And then you take them something that is truly cross-disciplinary and you fall between the silos. So the antibiotic discovery work we're doing, it involves a big collaboration. We have people who are specialists in the history of language and the history of medicine. who are helping us sort of source the ethnopharmacological texts and interpret them. We have data scientists who are trying to find patterns in those. We have people like me who are doing the microbiology. We have pharmacologists. And we should tick all the boxes of crossing all the disciplines. And yet everyone says, no, you don't quite fit. And so actually for the first few years, we only had success getting funding where we started to split ourselves up and hive off different bits of the consortium to different funding. And it's interesting because in the last couple of years, there suddenly has been a few sources of real cross-disciplinary funding. So a few of the different learned societies and the Leverhulme Trust brought out the Apex Awards, which are entirely for cross-disciplinary work. We were lucky enough to be part of one of those. But those schemes are very few and far between. And so trying to get the big chunks of work done in an interdisciplinary area has been quite difficult.
Interesting. I love the way you use the word lucky. We all use that word lucky, but actually it's because you were excellent. So maybe we should start using, we were excellent. I got that funding.
So that's the thing. And even if you missed, you were still excellent. You didn't quite tick the box on that day.
So Mariam, tell us about funding your work.
I was about to say I've been really lucky. Let's see. Cancer research is funded well compared to other disciplines. But even then, there's a huge amount of competition. And I think it's always been that the research question needs to be well defined. It needs to be realistic but ambitious. Some of these are in conflict with each other. Realistic but ambitious. It needs to address an unmet need. There needs to be a very obvious translational clinical relevance. There needs to be a very apparent benefit for mankind. So, the bar is set very high. And there are smaller and versus larger funding bodies. I've often worked with Cancer Research UK, and it's here where I truly think we've been lucky in that they very much so foster team science. So, where Freya was saying that there's been a shift towards multidisciplinary, I think in my field it's natural that you work across disciplines, from pure scientists to academic clinicians, to pure clinicians, to clinical research teams, laboratory staff, technical staff, and so on. So a lot of the work that I've been involved in has been, we use these terms longitudinal, they've been genomic studies, so that it's needed a lot of money, a lot of compute resource. So we've had to aim high. We've had to go to funding bodies that have been interested in collaborative team science. But I go back, and they've not always been successful. And at the moment, you start out with some smaller grants, smaller project grants, and you aim big. But aiming big, I find, is always good when it's a collaborative effort, because people will be at different stages of their career. And I think if you can bring different disciplines together, then surely the end product will be, I think, more fruitful. Not always. Sometimes too many cooks, as they say. But I think if it's a concerted effort, so you need like-minded people in terms of the drive and the desire to achieve, but certainly different backgrounds that bring together the research. So I think research and funding can be tough, and you need to be prepared for the hurdles and failures. And I'll always keep going back to the fact that you just need to be in a good team and a supported team. If you're going it alone, it must be really tough, I think, the way you described as, the 13 grant applications in year one.
Excellent. Thanks very much, Mariam. Jen, tell us about funding your lab.
Will do. So I'm in the lag phase. So I only actually got my academic post a year ago. And I've put in a few applications since then, and that's sort of given me practice. I got it's internal funding that's been allocated to the university by the Wellcome Trust, and it's got a competitive process, and I've been reasonably successful there, but they're all sort of small pots of money to get my pilot data. I was going to say lucky, but I'm in the point in my career where that means I can apply to, there's a as Freya was saying, the multidisciplinary thing is a big problem, because I can fit into the BBSRC for biological side of my research, the EPSRC for the engineering side, the MRC for the translational side. But whichever one you go to, I think sometimes the problem is the people who are going to be reviewing, even though they might have an idea of your side, they will be different to some aspects of it. And it makes it very hard for them to review it in the light that you're trying to present it. But the thing that makes me sort of lucky at the minute is that I am eligible for a UKRI, so the UK Research and Innovation, whatever, I can't remember. There is a scheme called the Future Leaders Fellowship. And actually that, because I'm early career, gives me a perfect platform to say, I need to do all of these things together. I'm not overstretching myself. I've got a record in this. And if you give me the money and the confidence for that amount of for, I think it's 4 years plus potential 3 more, I can get myself set up and I can have all of this pilot data, and I can have started all of these collaborations and brought in the clinicians, because one of the key things for me is, and the reason I went down the route of research I'm pursuing is because I met a friendly local vascular surgeon who told me about the problems in the limb salvage service. And that, you know, and I had a conversation with him and I said, I didn't think you'd ever want to talk to me. And he said, why? You do the science. I don't understand that. I just know what's wrong with my patient's feet. But I think in terms of funding, I've got all that to come. And I've spent, the benefit for me is that I've spent the last 6 months trying to put together this fellowship application. I didn't get past internal review in June, but I'm going to put it in January. Now, the benefit for me is that that's made me plan out all of the things I would like to do over the next 7 years, which means, I can't remember whether it was Freya or Mariam, I'm sorry, it was saying before about petitioning the research and putting a specific bit to a specific council, because I now have a big plan. If I put this in and it doesn't go anywhere, well, now I can see how those sections fit together, and I can almost pull bits out and say, okay, fine, I'll target BBSRC for that one. I'll target elsewhere for that one. The funding to do the translation is going to be even worse, but I'll cross that in the future if that's fine.
I think what you say, it's really good, because at we're all at different stages of our career, so the audience can get a flavour for what it's like when you start, and what it's like when you're flying high, and what it's like for someone like me, where I feel that I've done a lot and now I'm really focused on the translation. So we're going to move on to the next question. This is all about role models. We've always been told that most people who are working in these are quite competitive fields, so we look up to certain role models, and my role model was actually my PhD supervisor, Professor Sandy Florence is his name, and he was a role model for reasons that are not obvious. He was a very good supervisor, an excellent scientist, but he was also very kind as a person. So when I was working for him, as I told you earlier, I was a single parent with 3 young children. The oldest was 8, the youngest was 1. And so I was, all over the place in the lab, childcare, and all these kind of things. And I was very reluctant to tell him I was a parent. I didn't tell him, and it slipped out by accident. But, the moment he discovered I was a parent, I felt if I told him that, this was the early 90s, that's it. He wouldn't take me seriously, or he probably wouldn't hire me as a PhD student because I was on a studentship. But the moment he realized I was a parent and it slipped out by accident, he couldn't have done more for me to help me, get my career on track. So he is my role model. So tell me, Mariam, who is your role model?
Can I start with a question? Did he have daughters?
He had 1 daughter and 1 son.
I think that's really interesting. I'm not sure if that's common. I don't know if you come across colleagues and friends who've had a similar experience. I think there certainly are, and I've found that if there are many who have daughters, perhaps can sympathise a bit more. I think for me, I was really motivated to study physics early on because I had a great science teacher. He really fostered a passion for me, a passion for science, and I loved it because I wanted to be challenged, and I really liked maths. I love physics, and seeing how much he loved the subject really rubbed off on me. So I think my teachers at school, and then I think clinicians, the great clinicians who've had great bedside manner, who've had a really good clinical acumen. There are quite a few, so I won't name them individually, but I think those old school clinicians who were able to really encourage us to learn, I think they've been my mentors. And then in science, I think especially, I have a child, and talking about some of the difficulties and hurdles, family and looking after children and childcare, like you've experienced yourself, can be tough. It can be difficult. We find a way through it. So I think role models in women who have succeeded and have been realists. So they've not pretended that they did it all without any difficulties, but have expressed their difficulties and shared with you ways in which you can address those hurdles and overcome them. So I think women scientists in particular, who are, say, group leaders and have been successful in their careers.
Brilliant. Thanks, Mariam. Freya, if you can quickly tell us about your role models. Q&A.
It's funny because it links back to that, our internal view of ourselves and how others see us, and what you and Mariam were saying, because I look at scientists who have young kids, mainly women, and think, my God, I could never be that organized. How are they so productive? And that's one of the qualities I do tend to admire in other people. I think I have more of a mental list of particular qualities that different colleagues and friends have. So rather than having one role model, I'll think, oh, so-and-so is incredibly good at managing people or at diffusing a difficult situation, or this person, is very, very good at planning out their grant strategy, and sort of making a little pick and mix selection of which qualities to try and emulate from different people.
That's good. Yeah. And Jen, if you could quickly tell us.
Yeah. So I think there's bits and pieces. There's like my grandfather and my grandmother for my curiosity and my willingness to look like an idiot if it makes people smile. But my biggest role model at the moment is my previous boss, who did my postdoctoral, Professor Eileen Ingham. She's led our group for years, and actually what she taught me was, first off, you can do great things even with hurdles. Secondly, maybe I don't want to be that good. Maybe I'd like more of my free time to do role-playing games. But actually what she's really taught me is I can get to where she is whilst dancing around my office and being unsure of myself and all of these things. And you can get there, and you can get to that level with those. The important thing for me is you can get there with those things. They don't discount you. They're not going to push you out. You just have to find ways to deal with them. And I think that's the biggest thing for me is she's got to professor of a huge group. She's got patents all over the place and all these things. And she has the same insecurities and things as I do.
Fabulous. So now we're going to go to our Q&A, and we've got a few questions. So I've got one from Ivy Alexander for you, Mariam, and Ivy wants you to just talk about metastatic disease and what that means.
So often patients who have a diagnosis of cancer might present with advanced disease. So the cancer from where it originates has already spread to another site in the body. So there might be a patient who presents with a tumour in their lung, and that's a lung cancer, but it has already sadly, say, spread or metastasized to the brain or the liver or the bone. So when we talk about metastatic disease, it's disease that has spread beyond the site of origin. And in understanding the process of metastatic disease, we want to know what it is about the behavior of the cancer, the cell, the genetics, and so on, that facilitates that spread. And equally, why is it that some tumors don't spread and stay localized? So these are the things that we're interested in. Not all patients present with advanced disease. Some patients are diagnosed early on. They can have curative surgery, or their disease can be controlled and we can prevent the spread or slow it down. So there are different stages at which cancers can present.
Thank you very much, Mariam. And Rachel O'Sullivan wants to know from you, Freya, whether you use organoids in your study.
So very topical question, because I've been having several conversations with a new collaborator about this recently. So we're starting to talk about what we could do in that area. And interestingly, the starting point for this is because our work with the ex vivo pig model is at such a point, we can actually start to benchmark new organoid models by seeing how they perform relative to the pig lung model. So we have for very key bacteria, we know what genes they switch on and off when they go in this model, and we know that correlates very well with what happens in a person. We know that the structures they make of the multicellular biofilms are very similar. So when people start to look at organoids, they can make some comparisons with what we're seeing in the ex vivo model. And then it's a case of picking the best model for the question you want to ask. So one downside of organoids is they're expensive, they require very specialized tissue culture facilities, but they can give you a depth of detail about host immune responses and host cellular responses that you can't get in any other lab or animal-derived model. So I'm very excited to see what we might be able to do with organoids, particularly in terms of what the hyperinflammatory immune response in a person might be doing to a pathogenic bacterium.
Brilliant. And I've got Bimal Decker asks, can organ-on-a-chip technology really revolutionize drug discovery? I think this is one for you, Freya, again.
So, yeah, again, this is a bit of a weasy answer. I say yes and no. I think if you're thinking about bacterial infections, right, there's kind of two big categories of bacterial infections. There's simple ones where a healthy person accidentally comes into contact with something that gets in through a scratch or whatever. You have a short-lived acute infection and it will respond very well to a drug. Now, in that situation, the bacteria grow in a very simple way, essentially. And the way they'll respond to a drug that's been tested in an organ on a chip might correspond very well to what happens in your body. The problem is when you come to the kind of infections I work on, and they, like in the CF lung and the diabetic foot, the bacteria stick themselves together in this multicellular structure called a biofilm. You can't really grow those realistically in the lab, and organ on a chip won't really predict what the bacteria do. So I think we need a sort of a fast, high throughput diagnostic sort of technology for simple infections, and a more sort of slow, artisan, bespoke testing methodology for these very tricky biofilm infections.
Yeah, cool. And I've got Ivy Alexander asking one other question, and this is for you, Jen, and saying, you know, there's an increased awareness of the medical sciences now, and again, it's just talking to funding as a result of the pandemic. And do you find, or do you think that there'll be more science funding in the medical sciences area as a result of our pandemic experience?
I mean, at the moment, from what I've seen, there's been a huge drive for funding related to COVID, which is great for all the people who have any research that could possibly have anything to do with COVID. And I think, yeah, people are, I wonder actually if one of the things that might come out is that we're realizing that a lot of the problems that come out are to do with people thinking scientists are these big secretive people who don't know anything. And one of the things that I hope will come out of this is more funding for us to go and talk to people about the research that we do. On top of funding, I think the pandemic is perhaps destabilizing everything enough that funding might be shaky for a while we get back to dealing with the current problem at hand, pushing really into that, finding innovative new ways to fund things for that, especially across the disciplines and across the world. But I really hope that actually something that will come out is an increased awareness of the importance of us talking to people about our research, demystifying it, making it more accessible, so that when we start trying to say things like, okay, you have to wear a mask and wash your hands, people perhaps understand why we're asking them to do that, and that it's not some secret, sinister thing going on or anything like that. And the same can be said of things like Freya's talked about having to use, you get to a certain point where we have to use animal models. There's no way around it, because we don't have anything that replicates a fully functional immune system. We'd all, I'm sure, I certainly want to get away from that if I can, but we can't. And I think, again, it's that conversation, being able to be open about what we're doing and why we're doing it, and give people information to make a decision that they understand and they can see what we're doing.
Yeah.
The question, but never mind.
Yeah, no, I mean, it is quite important to really make people understand that the fact that there's more research funding, it's actually research funding for COVID, and it may not affect me making a pain medicine, you know. But Karishma Kulshik has a very interesting question, and that is, should we be so open about the challenges around working as a woman in a STEM in a STEM career. Do we think that being this open will actually challenge the stigma associated with, you know, us feeling a little bit insecure, or is it going to frighten people away? I mean, I'm going to ask you, Jen, what do you think about this? Because
absolutely, I think the reason I have got where I am is that when I started to have the conversations with my colleagues and I started to realize that I wasn't on my own, that this is, I'm not going to say this is perfectly normal. This is a rubbish way to be. There are terrible things in our careers that make this really hard. But I think realizing that a lot of people around you have this, and they're not all women. So there are a lot of men who have imposter syndrome. I'll put the term out there, who have the same problems, but they don't necessarily talk about it as much. And I think being able to do, you're going to stop.
Yeah. Thanks, Jen. So thank you very much for all those that are listening. We've come to the end of our time, and thank you very much to everyone, to Freya, to Mariam, to Jen, for really being such fantastic panelists. And goodbye.
Thank you.
Thank you very much, everybody. Thank you for watching.
Bye.
And thank you to our panelists.
Bye-bye.
Bye.